Welcome everyone to STEM Talk session 20 of the 7th Polygen Symposium of Rising Scholars. My name is Jin. I'm one of the founders of Polygens, and it's always a pleasure to be able to see and celebrate all of your hard work with you all in each and every symposium. I will be moderating today's session, and the goal of the symposium, as we all know, is to celebrate and showcase all of your hard work. And today we have 4 students presenting on a variety of topics, and a fellow member of the Polygens team will be scoring your presentations, which will help us determine prize winners after the whole conference is concluded. If you have any questions, and I highly encourage presenters also to ask questions to each other, type them in the Q&A, and then we will have time at the end of each session, each presentation, for each speaker to answer some of the questions that have been asked. And so today's order, we'll start with Myra, and I'm super excited to have her share her screen and present, and I'll spotlight her for everyone, and then if everyone else could mute and turn off their videos, that would be great.
Okay, I'm going to share my screen real quick. Okay, can you see my screen?
Yes, all good.
Okay. Okay, so hi, my name is Myra Clark. I'm a sophomore at the Thomas Jefferson High School for Science and Technology, and my project is mental imagery and social phobia. Which form of imagery-based cognitive behavioral therapy is most effective? So the research goal was to investigate which form of cognitive behavioral therapy is most effective for treating social phobia. And to conduct this project, I reviewed current literature on CBT and social phobia, and I wrote a research paper about this topic. So here are some terms I'll be using during this presentation. To start, social phobia is a disorder classified as a fear of social situations, and it involves imagery portraying someone experiencing anxiety symptoms. Mental imagery is the internal experience of representing sensory information normally perceived through the senses, but entirely in the mind. And it's often referred to as visualization. And cognitive behavioral therapy, which is abbreviated as CBT, is a form of therapy which attempts to reduce symptoms of psychological disorders. So why is this project important? There hasn't previously been research done on the effectiveness of different forms of CBT in treating social phobia. So the purpose of this was to focus primarily on this disorder. So now let's get into some background information. According to previous literature, there are 2 types of information processing in the brain. There's top-down processing and bottom-up processing. Mental imagery is a form of top-down processing, which involves higher level cognitive processes, which are more complex and conscious processes, such as mental imagery and spatial navigation. Additionally, in this type of processing, in simple terms, information moves from cortical or like outer areas of the brain to inner areas of the brain, hence the name top-down. And bottom-up processing, on the other hand, is the exact opposite, as information moves from inner areas of the brain to cortical areas of the brain, and it involves lower level or less conscious processes. In fact, there are 2 cognitive processes that are very similar to mental imagery, and they are called sensation and perception. Mental imagery is actually often referred to as a weak form of perception, which can be defined as how someone thinks they're perceiving something rather than how they are actually perceiving it. And although these are similar, perception is actually more related to imagery because they're both top-down processes, whereas sensation is not. To provide evidence that mental imagery is a top-down process, research has shown that there's elevated activity in the prefrontal cortex during imagery and decreased activity in the visual cortex, or an inner area of the brain. Imagery not only plays a role in treating psychological disorders, but also in the symptoms of these disorders. For example, PTSD, or post-traumatic stress disorder, involves involuntary flashbacks or mental images portraying a distressing event. Additionally, imagery produces a greater emotional effect than a verbal representation of a word, which has been shown through research. So my project focused on 2 main papers, but I'm only going to focus on one during this presentation due to time constraints. So one of the main paper that I investigated ranked different studies investigating the effectiveness of different types of CBT based on promise and the outcome of each study that was investigated. And I will talk about these subtypes on the next slide. So before moving on, I would just like to point out that there are other subtypes of CBT than what I'm showing on this slide, but these are just the most relevant to the presentation. So imagery rescripting is a type of CBT which involves imagining an image and then changing the meaning or emotion associated with that image. And it has many benefits, including it being effective. Oh, I'm sorry, I didn't mean to do that. Including it being effective in both trauma and anxiety. And it involves interaction, which is manipulating imagery and consciously feeling as though you're there. And I'll discuss this more in a later slide. So guided imagery is imagining positive images while being guided through the process, and it can have variations of images. And it's beneficial because of the mindfulness-like state, and it could be beneficial because of the calming technique, because it's calming rather than imagining distressing imagery. But this means it doesn't target the source of distress, which can make it less effective because it's not really changing how the person reacts to a triggering situation, and it's only a technique to calm down. Positive imagery is imagining positive images while being guided through the process, and it can also have variations of imagery, so it's similar to guided imagery. And this means it could have similar benefits and drawbacks to guided imagery. And next is imaginal exposure and systematic desensitization, which are imagining a feared image that triggers anxiety and stress, and systematic desensitization is a more gradual approach. Imagery-enhanced protocols combines imagery rescripting with positive imagery, and then seeing is not believing is a method which was suggested by a paper that I read, and it involves realizing that imagery is not reality. And then finally, traditional methods involves being exposed to a real-life fear, and it relies on bottom-up processing. So given the research, I think imagery rescripting is the most effective method of CBT because it involves interaction with imagery. And additionally, imagery interaction likely involves top-down processing, which, as stated previously, has direct contact with emotional systems in the brain, and imagery manipulation produces a greater emotional effect in therapy. Although I believe this is the most effective singular method, imagery-enhanced protocols seems to be the most effective combined method, combining positive imagery and imagery rescripting. However, I can't really accurately determine which one is more effective. And lastly, the seeing is not believing method could also be very effective. So you may be wondering what exactly is interaction. So it's an important term used to describe some CBT methods, and it involves two main things: conscious manipulation of imagery or changing it in some way, and feeling like you're a part of the image. So it's really important, as this conscious manipulation of imagery is a top-down process which has direct contact with emotional systems. And for example, this would be similar to actually giving a speech. Imagery interaction could be very similar to actually giving a speech versus watching someone else give a speech. Interaction would be like giving a speech because you're consciously manipulating what's happening and you're actively there. But then watching someone else give a speech, you're really just watching it happen. So I believe that CBT could be more effective than traditional methods because it relies primarily on top-down processing and conscious manipulation. And traditional methods rely primarily on bottom-up processing, which can produce less of an emotional effect. So some future research recommendations include investigating whether singular methods or combined methods of CBT are more effective, comparing how effective different treatment methods are in treating social phobia. Could CBT be combined with traditional exposure methods and what effect that would produce, and further investigation into the seeing is not believing method. So thank you all so much for listening to my presentation. Here are my references, and thanks again.
Thank you so much, Myra. I personally suffer from arachnophobia, so fear of spiders. So this is super helpful in terms of thinking about how to help myself. And I was wondering, how I think in popular theory, a lot of people think that, oh, you should just expose yourself, just be in a room with spiders, or the next time you see spiders, hold them or touch them, or some therapists use that method. From your research, how do you view that kind of just face it, or force yourself to interact with not just the images, but the actual thing you're afraid of? What are your thoughts on that?
So first of all, I think that's kind of not necessarily the best way to approach it. To just telling someone to just face your fears, that's not really an effective method. And most people, I don't think, would be really willing to do that. But also based on research, I think because it's a form of bottom-up processing, or sensation, feeling like you're actually holding a spider, for example, I think it wouldn't be as effective because you're not really controlling what's going on, and you're kind of in this state of shock, And I mean, it does technically desensitize the person to their fear, but I don't think it would be as effective.
Compared to CBT, you mean?
Yes, compared to CBT, sorry.
No, makes sense. Great, awesome. Thank you so much. For time reasons, I'm going to move on to Akshat Ubale's presentation, but thank you so much, Myra. That was awesome. So
thank you.
Yeah, Akshat, feel free to share your screen, and everyone else will mute and turn off camera.
Okay.
All right, thank you. All right, I'll start it out. So my name is Akshat, and I investigated CTCF factors and their influence on GI cancers. All right. So my main question when I was investigating was how and where do CTCF binding sites change over time in the gastrointestinal system? And so first, before I move on, let me define some key terms. Gastrointestinal cancers, otherwise known as GI cancers, are cancers that develop starting in the GI tract, which are organs spanning from the esophagus to the anus, as seen on the diagram on the right. And they have to start in the GI cancer, but they can become metastatic and spread throughout the rest of the body. The key thing here is that it starts in the GI tract. Mutation hotspots, otherwise known as just hotspots. First, let's start with mutations. These are mutations or modifications to the nucleotide sequence or DNA, and some mutations can cause tumors. Now, this is important as mutation hotspots are locations in the genome where amino acids have been mutated a lot. Otherwise, mutations happen here again and again throughout many cases and has a high mutation frequency. And this is important as it can suggest a potential mechanism that causes mutation at that same spot throughout many genomes. Some more definitions is tumors, which is a massive tissue that forms when cells are experiencing a lot of division that it's not controlled. That's usually due to mutations. Transcription or binding factors, those terms go hand in hand, are proteins that regulate the expression of certain genes by binding to the gene's nearby regulatory sequences. An example of this would be CTCF. A DNA binding site is a sequence of nucleotides where molecules can bind, and these are mainly used by binding factors, and these sites differentiate themselves to their sequence. Cohesion complexes have many functions, including DNA repair, and they also regulate gene expression by arranging chromatin, which is just DNA, into large topologically associating domains that separate large chromosomal neighborhoods. Basically simplified is that it organizes DNA, and such proteins work alongside CTCF. What is CTCF? It's just like the cohesion complexes, CTCF separates local chromosomal neighborhoods and arranges them into TADs, but it also organizes short and long range contacts with these domains, and they're moderated by CTCF. And here's the structure of the CTCF protein. So now for some background, GI cancer mutations mainly present themselves in the stomach and the colon when it comes to CTCF mutations. Dr. Kordmansky from Yale Medicine found that colorectal cancers seem to be the most common form of GI cancers, but then when we look at CTCF binding, we find that 25% of GI cancer tumors and 19% of colorectal cancer tumors are mutated at a CTCF binding site. And this is relative to other forms of diseases involving CTCF binding site mutations, showing that CTCF mainly presents itself through cancer, GI cancers. And then we also some more background is that mutations accumulate between the 30s and 50s. In this red box, we can see that between the age of 20 and 39 and 40 and 59, there was a significant spike in mutations in those age groups, showing that when you get to around that age group, you start experiencing a lot of mutations in your body. Now onto the hypothesis. CTCF binding site mutations accumulate between the 30s and 50s of one's life and mainly present themselves in the stomach and colon. Now, we talk about how the 25, 29, 19% data, but there is some more data to this. That these mutations do accumulate on CTCF binding sites. It's not a correlation, but it's in fact causation. Past studies have indicated that these binding site mutations actually drive GI cancers, and that of this 25, 19% data we found, each one of these had a mutation in one of the 11 CTCF identified binding site hotspots. And then 3 of these hotspots were correlated with a change in expression of CMPQ, KNCQ5, and SPG20. Knowing this is important, as if we can investigate the expression of these genes or the expression of these genes, we can help explore our hypothesis. So for materials, we use CTCF transcription factor chip sequencing data from the ENCODE database for all these following body parts, and then we use the USSC genome browser to find the location of these genes seen on the right. So for results, we found that every body part experienced more differences than similarities across the 30s and 50s, but that specifically the stomach, the gastroesophageal sphincter, and the pancreas had the most amount of differences and also the smallest amount of similarities. And then we found that the transverse colon, the esophagus muscularis mucosa, and the esophagus squamous epithelium had the biggest amount of similarities and also the smallest amount of differences, showing that most mutations or differences in binding accumulate in the stomach, the gastroesophageal sphincter, and the pancreas. We also found that there was no change in the binding on the genes of CMPQ, KNCQ5, and SPG20. What does this mean in simple terms? Is that between the 30s and 50s, there was no change on these genes, meaning there's no change in the expression, theoretically. And so this was contradictory to the evidence that we found in past studies. Now, what this data actually means is first that between 37 and 50-year-old, most people will develop GI cancer-related mutations in the stomach, gastroesophageal sphincter, and the pancreas, but not the colon, which was surprising, which means either the colorectal cancer doesn't present themselves through CTCF mutation hotspots, or they do so, but not at the ages we investigated. But gastric cancers do. Gastric cancers, by the way, are another word for stomach cancers. Now, the importance of this research, assuming it was the universal truth, there are some flaws, but we'll get into that later, is that with further research, these findings can give healthcare professionals a guideline on where and when to look for GI cancer developments, but it can also help cancer researchers know where and when to look for potential interventions, preventions, cures to these related cancers through this CTCF transcription factor. Some potential research that can stem from this is that research of when an adult is likely to start developing these cancers and whether or not it's unique to the 30s, 50s, meaning can it happen the 50s and 60s, 60s and 70s, earlier on? More research can help solve this question. Research on what percentage of colorectal cancers come from CTCF mutation hotspots can help further explain findings, because we do know that colorectal cancer is the most common form of GI cancers, and so there's still hope that it can come from CTCF mutation hotspots. We just need to do more research. We've barely scratched the surface with this project. So my unanswered questions was, why didn't we see a change in the binding of CMPQ, KNCQ5, and SPG20, despite past studies saying so? Is that we had differences in studies. The past study measured the change in these genes through expression, while we measured through the change in binding. The issue with this is that binding can occur outside of the gene, but affect the gene, meaning if we truly wanted to measure the grand scope of the gene's change in binding, we have to measure those regulatory sites, which we may not have done. So in conclusion, again, our hypothesis was true to the fact that there was a significant change in binding and more mutations, but they weren't related to the colon, but in fact, the stomach and some other body parts, and they weren't correlated with the genes we expected them to be. Now, onto some limitations and some limitations on this data, because not every research is perfect. First was a lack of trials, but also a lack of body part specific sequencing. This whole experiment was actually based off of 37 and a 50-year-old sequencing data, and that only 2 people, and then we also didn't explore the entirety of the GI tract. We didn't go through the liver, the mouth, and other body parts. And this is all due to the fact that this data was just simply not available to the public or online, which was what my availability was constricted to. And so I guess in future would be using more data, more types of body parts would be helpful in making more accurate and fruitful results. But then also a lack of context. When I went on ENCODE and got this 37 and 50-year-olds data, I didn't know why this data was originally collected. For all we know, these could be pancreatic cancer patients, and so that could explain maybe why we saw a lot of changes and mutations in the pancreas, right? There's no real context, and that wasn't available. So in the future, having access to this context on this data would be very helpful in making sure the data we use is valid. And so here are my references. And thank you for listening. I'd like to thank my mentor, Vera, for helping me a lot through this project. And if you want to work with me or ask any questions, you can reach out to me or ask a Q&A here.
Thank you so much, Akshat. Super interesting. I was wondering what made you excited to pick GI cancers as your topic? There are so many different cancers in the world to choose from, and I was wondering what gave you that spark to pursue this?
Right. So initially starting with this project, I actually just wanted to work on transcription factors. And so I found this CTCF transcription factor I was interested, and I found it a correlation with GI cancers. And just based off standard knowledge at the time that I had, I knew colon cancer was a huge thing, like very common, especially when it came to like when you got older. So I just thought like a lot of stuff was aligning. Why don't I base my research off that stuff?
Cool. And we also had one anonymous question from the Q&A, which is, what makes a cancer more deadly than others? So we generally know, for example, that if you get diagnosed with the cancer in stage 1, it's usually easier to solve the problem than if you get diagnosed in stage 4. But there are some cancers that are smarter at staying hidden until later. And I was wondering, is that also something that's controlled by mutations or genes, or is that really just depending on which organ it affects?
I think it's a variety of factors, and I think I addressed part of it when you said hiding. That's kind of true, right? CTCF, like when we try to like diagnose cancer, there's many ways, but this could propose a new way, right? For example, right now cancers could be hiding through CTCF factors. And so since we don't know that, the cancers could develop more and more. So potentially identifying through CTCF could help that. But on how cancers get worse and worse, it starts off as just a mutation, and then a tumor develops. And now if that tumor spreads to other parts of the body, becomes metastatic, that's where it becomes deadly and like almost incurable. But if it's just a tumor, it's not even considered a cancer. They just remove the tumor. But hopefully that answered that question.
Great. Thank you so much. Awesome. We'll move on to the next one just to make sure that everyone has enough time. So thank you so much, Akshat. Next, we have Manasvi, and I'd like to invite you to share your screen and kick us off.
Can you see my slides? Okay.
Yep, we can see it.
So, hi, I'm Manasvi, and my Polygens project was focused on answering this research question. How can the lung microbiome be used to predict disease severity in a cystic fibrosis patient? So, cystic fibrosis is a fatal genetic disease that is caused by a mutation in the CFTR gene. And there are actually expected to be more than 160,000 cases of CF worldwide, including undiagnosed ones, and about 1,000 new cases of CF are diagnosed each year. In general, this disease is commonly known to be a progressive lung disorder, but it does affect many other organs, including the pancreas, the liver, and the intestines. So nearly every single CF patient is expected to experience lung infections as their disease progresses, and approximately 80 to 95% of patients experience fatal respiratory failure that is caused by chronic lung infections and inflammation. So if we can track disease progression by looking at the lung microbiome, we can guide doctors with their treatment plans for patients by prioritizing what microbial type needs to be targeted and what antibiotic needs to be administered at a certain time point. So in my project, I used 16S sequencing in order to determine the lung microbiome composition of the collected samples. And by lung microbiome composition, I basically mean the types of different microorganisms and their abundance in the lung. So sequencing in general is just the reading of the order of nucleotides in a sample. And in the case of 16S, we can compare the sequence that we read against publicly known sequences to figure out what microorganisms are in the sample. Since the 16S gene is universal in bacteria, the presence of variable regions of that gene can then be used for taxonomic classification. So from this figure, after DNA isolation and after PCR amplification, where certain primers target the 16S gene, the sequencing part is where operational taxonomic units are formed based on how similar these sequences are to the known reference sequences. So my hypothesis was that dominant CF pathogens such as Pseudomonas aeruginosa and Staphylococcus aureus which have been identified in past literature, would have a strong negative correlation with FEV1 score. And this is forced expiratory volume in 1 second, and it's considered an effective measure of lung function in the medical field. So before this, a quick note on the data set I used is that it came from the CHEETAH website, and it consisted of 51 samples of the lung microbiome from CF patients, and the link is at the bottom of the slide. So from the raw data, the first step was to clean it up and then to classify the sequences. So we first filtered by length. So anything longer than 275 base pairs was removed. Then anything that had too many unidentified base pairs was removed because we didn't want too much uncertainty in the classification process. Then to maximize efficiency, we deduplicated the sequences, which basically kept only the unique sequences. And then sequence alignment made sure that these sequences could even align with those for microbials and not some other type of organism or species. And this was done by making sure that the sequences aligned with the V4 region of interest on the 16S gene. So next was pre-clustering. So only sequences that had 1 or 2 base pairs different were grouped together. And with chimera removal, we were able to remove any incorrect mixes of 2 or more biological sequences that were formed during PCR amplification to make sure that we didn't identify any of the sequences as some other novel organism. And after this was taxonomic classification. So from the taxonomic classification, we got 4 main outputs. So the first was sample metadata, which was basically sample information, notably the FEV1 and FVC scores for each patient, which are basically measures of lung function. The second was a representative sequence table, which was a clustered data matrix of the different sequences for each patient and their corresponding representative sequences. The third was a count table, which contained the number of times each representative sequence had corresponded to each patient. And the aggregated sequence table was basically a combination of the representative sequence table and the count table to display the total counts of each of these sequences for each patient to make it easier for the statistical analyses. So in the statistical analyses, I did a Bonferroni correction to account for multiple hypothesis testing. So what this basically means is that in order to identify the significance level for the hypothesis test, I did 0.05 divided by the number of tests performed, which in this case was the number of bacterial clusters, to get a value of about 0.0004. Then the Spearman rank correlation was used to determine the relationship between bacterial clusters and FEV1 scores. So here's an example of one of the results I got. And just as reference, the R-squared correlation coefficient shows how the strength of the relationship between the 2 variables. So 0 is no correlation, and with 1 being the strongest correlation. So with Fusobacterium specifically, we can see that as FEV1 score increases, we can see a positive and moderately strong relationship between the 2 variables. And I did not include the other top hits that I got in this presentation for the sake of time, but they can be referenced in my paper. So according to past literature, the core members of the general airway microbiome were the following, and we can see that Fusobacterium is included in these types. So this supports the idea that as the disease progresses, the core members of the general airway microbiome are being displaced and being replaced by top CF pathogens, such as Pseudomonas aeruginosa and Staphylococcus aureus. And it was quite interesting that these were not identified as top hits, even though a number of research papers from the past often identify these as top CF pathogens, which is why I chose them to be a part of my hypothesis. So one limitation was that I had sparse data. And this basically meant that I had many zeros in the count matrix, which showed the counts of each representative sequence for each patient. And the problem with this is that it could hinder the analyses on data after classification because of poor performance from analysis algorithms. And the second limitation is the reliability of the FEV1 score for lung function, because past literature had shown that biometric information like BMI had been shown to influence these scores when used as an indicator for lung function, and there was no indication in the data set or on the website of whether or not these scores had already been adjusted to factors like age, height, and weight. So we can't exactly determine the reliability of these scores when trying to determine correlations with bacterial types. Finally, one future direction would be to identify microbial types that are correlated with lower FEV1 scores as well, because most of my results mainly focused on identifying top hits of microbial types that were positively associated with FEV1 scores. And this could be done mainly by looking for more data sets with more samples for further analyses, essentially. And finally, another question I would like to investigate in the future would be of whether microbial diversity in the lung decreases as cystic fibrosis progresses, because this would allow for better determination of which dominant bacteria type we need to target using treatments or antibiotics at a certain time point. Thanks for listening.
Thank you so much, Manasvi. I had a quick question about how and whether your experience in this project has affected what you're thinking about pursuing later. So do you think that, for example, looking at cystic fibrosis and gene sequencing is something that you would want to take beyond high school and into college as well? Or what have you learned from this experience in terms of how has that informed your future thinking?
I mean, definitely. The thing that actually inspired this project in the first place was my work on CF from before. So I was volunteering with the Cystic Fibrosis Research Institute and working on their awareness campaigns in the South Asian community, because the disease is actually underdiagnosed in other populations other than like the Caucasian population. So that's what got me started with this project, and including my interest in genetics as well. So because of the fact that cystic fibrosis patients, even now, their like average survival rate is only until about 50. I think there's a lot more research that needs to be done. And I think this project has pointed me in the right direction of like for the research, and could definitely impact what I'm going to be doing in the future.
Amazing. And we had another question in the Q&A about what during this entire process was the most challenging. Was it the statistical analysis? Was it defining the scope of the project? Or was it some other aspect of your experience throughout the 10 sessions?
I think the most challenging part would probably be staying organized with microbiome data, to be honest. It was my first time working with all these sequences, and it was really hard to figure out in the beginning how to like go from these random sequences of variations to the classification step. And on top of that, we had to like do our own aggregations from like the representative sequences to the total counts. Like they weren't readily given by the pipeline. So it was kind of difficult to figure out, is this what I need to do the analysis on, like on the representative sequences or on the total counts? It was kind of hard to put everything together, but at the end it was worth it.
Amazing. Thank you so much for sharing. Awesome. So let's move on to the next presenter. Thank you so much, Manasvi. Last but not least, we have Krithin. Super excited to hear your work. So feel free to share your screen and get us started.
All right. Can you share my screen?
Yes.
Okay. Okay, so my presentation is on a literature review on the clarity perception enabled autonomous vehicle safety applications by me, Krithin Kamineni. All right, so the goal of this literature review is to provide important insight into the future of connected vehicles or autonomous vehicles. Along with various emerging technologies reviewed in this paper, each comes with its own evaluation of limitations related to this technology. So limitations of these technologies need to be evaluated, since these technologies are the line between an accident and a prevention of an accident. Readers of this autonomous vehicle technology literature review will gain a thorough understanding of emerging applications and how they can improve safety. So how do connected vehicles operate, or autonomous vehicles operate? So to start off, autonomous vehicles, or autonomy, is a method in which a vehicle is capable of performing safety maneuvers on its own with the help of other on-road vehicles. So first of all, V2V technology, abbreviated as V2V, is a method of communications between on-road vehicles, which helps improve the safety of passengers in those vehicles. Through V2V, vehicles can exchange safety information like speed, location, which help vehicles detect potential crashes and alert the driver. So the next technologies that autonomous vehicles use is clouded perception, which uses key information from sensors mounted onto vehicles and the transferring of that information to other vehicles. This also relates to the processing of this data into a clear understanding of and vision of the world around the said vehicle. With the use of both V2V and clouded perception, autonomous vehicles are capable of executing safety applications like lane changing, collision warnings, and left turn assist. So the steps to execute safety applications. So the first step is availability of information. The initial step in this process is in which AVs or autonomous vehicles collect information from other vehicles and they use it through the next step. So this is availability of information. The next step, step 2, is localization. So localization provides the vehicle its exact position in the cloudly produced map with reference to static or other dynamic objects in its surroundings. And last but not least, the third step is safety applications. So safety applications are the actual maneuvers that autonomous vehicles perform in response to obstructions in the clouded perception messages. So next, let's take a look at each of these steps more thoroughly. So step 1 is availability of information. So before an autonomous vehicle incorporates clouded perception to perform and execute safety applications, it must go through this initial step of realizing information and effectively using it to prevent an accident. So this step has to do with the number of vehicles exchanging key information using clouded perception. All of these vehicles use this exchange information to spot potential collision threats and any other situation in which the passengers are at harm. The papers that are reviewed in this section of the literature review explain how this information can be reduced and how exchanges are passed as valid and safe. So the next step, step 2, is localization. So localization in autonomous vehicles helps provide the vehicle its exact position in the collected map or on a road map with reference to static or dynamic objects surrounding it. This process of localization provides an accurate location and allows the vehicle to understand its environment and any obstructions in it. Autonomous vehicles and clouded perception-enabled vehicles are also able to capture their trajectory as they move along the road. This allows these vehicles to better respond to potential dangers. And the next step, step 3, is safety applications. So safety applications are the maneuvers that autonomous vehicles perform in response to clouded perception messages from other vehicles. These applications use the localization of the vehicle and the exchange information from the surrounding vehicles in step 1 to perform these safety applications. So safety applications are the final step in autonomy, as they use all the collected information and every other previous component or step to perform one action to prevent a collision and any harm of a passenger. These applications surround a variety of maneuvers like lane changes, braking, lane centering, and much more. So next, let's talk a little bit about future technologies. These technologies are not possible right now because of their extent, but are possible in the future as technology advances. So the first technology is a brain-controlled vehicle. So the brain-controlled interface, or BCI, is connected to an autonomous car and used to perform maneuvers in response to physical threats. So the vehicle, in simple terms, is controlled by a human brain, which is able to allow the vehicle to perform maneuvers in response to threats. The autonomous vehicle in the system is equipped with a variety of sensors like cameras, LiDAR, motion, and much more, and can be controlled by a computer based on the BCI. The next technology is a driver alert system. So this system provides an alert to the driver of the receiving vehicle based on data of the characteristics of the sending vehicle. This data is based on characteristics of a sending vehicle and map data for different terrains in which both the receiving vehicle and the sending vehicle are located. So this is an image of the first technology. And last but not least is key takeaways. So key takeaways, I present a literature review that provides insight into the future of connected AVs or autonomous vehicles. So supporting technologies are V2V, which is vehicles exchanging information like speed, location, which help the vehicles detect potential crashes and alert the driver. Next up is clouded perception, which has to do with the collection of key information from sensors mounted onto vehicles. So next are the 3 steps for providing collaborative perception to autonomous vehicles. First off is availability of information, which allows vehicles to realize information and effectively use it. Step 2 is localization, which provides the vehicle its exact position in a map or a roadmap with reference to other objects or vehicles in the map. Last but not least is safety applications, which are the actual maneuvers that autonomous vehicle perform in response to collaborative perception messages. And last but not least, in this presentation, we reviewed some future and advanced technology like the brain-controlled vehicle and the driver alert system. And that's it. Thank you for listening. And I just want to say a big thanks to my mentor, Noah, for always helping me throughout my project and guiding me throughout it. Thank
you so much, Krithin. Super exciting. There are so many startups and companies, big and small, working on the same technology. And I was wondering, so based on your research of all the different types of technologies that go into the creation of self-driving or autonomous vehicles, do you and your mentor like have a prediction of which company will make it first in terms of like reaching that first level of, I forget what the phases are, but that like phase of autonomy where it can truly be driverless? Do you have any sort of predictions or thoughts on that?
So like this research like on technologies is done by like individual research institutions, so not like actual car companies. But then after those technologies are like passed as like valid and safe, then like car companies like adopt those technologies. But I personally think, I mean, obviously like companies like Tesla and like other, more like luxury car companies, they have more like funding to fund those technologies and the improvement of those. So I would say like probably Tesla, because they already have like an advanced system to drive without a driver.
Got it. There was also a question in the Q&A about how autonomous vehicles can detect the traffic lights, especially when there are often many sets of traffic lights at the same crossroad, for example. How does the vehicle know which set of traffic lights is theirs, if that makes sense?
So vehicles, they use different sensors, like some use cameras, some use what's called LiDAR, which uses lasers to detect like objects in its map. So most of the vehicles that detect like traffic lights, they use either cameras or LiDAR, so that they can detect whether it's like green or red or yellow. So LiDAR pretty much uses lasers to pretty much like detect those signals, and then it bounces back to the car to like process the information. And yeah.
Got it. Super exciting. Cool. Great. Since we have actually a couple minutes left, I'd like to invite all of our presenters to come off mute and turn off their cameras. And I have a couple of just discussion, reflection questions that I'd love for us all to reflect on, unless there are other questions for Krithin or other questions that you guys have for each other after listening to all of each other's presentations. Any questions or reflections?
Oh yeah, I actually have one.
Yeah, go ahead, Akshat.
Krithin, I'm not too sure if you went over this, but I was wondering personally, which technology do you prefer like the most out of the ones you went over?
So we went over like a lot of technologies, but I would say probably the safety applications, because they actually provide like a physical maneuver that prevents an accident. So that's like the most like applicable technology in the real world. So yeah.
Thanks.
Awesome. And I wanted to ask everyone the same question that I asked Manasvi, which is, how has the experience in doing this project changed or affected what you think you might major in college? And I know some of you guys are maybe seniors or juniors starting to think about college and college majors. I'm just curious how you think your Polygens project fits into that larger path, or was your Polygens project for you really like a side project and something you were very curious about, but not necessarily as like a potential major? Myra, you want to go first?
Sure. So wait, I'm sorry, I don't have my earbuds in anymore. So can you hear me?
Yes, we can hear you.
Okay. So I've already like had an interest in like neuroscience, psychology, like that kind of area. But I think this made me a lot more interested in like both neuroscience and psychology. And so I'm a sophomore in high school, so I'm not entirely thinking about like my college. I mean, I think I want to major in neuroscience, but I'm not there yet. So, but my school does offer neuroscience as a class. So I'm thinking I want to do that as like a senior lab project.
Cool.
But yeah, I think it's really made me a lot more interested in neuroscience.
Awesome. Cool. Any other thoughts and reflections from Krithin or Akshat or Manasvi, if you have new thoughts? Yeah, Krithin, go ahead.
I just want to say, so before starting the project, like when I first started, I just thought of it as like a side project, but as I moved along, I became like a lot more interested in what I was doing. And I already wanted to like major in CS, but like now that I looked into like autonomous vehicles, like that's just like a different application for CS. So that's something I can like branch out into after college. So yeah.
Totally. And I think it's a field that where there are still so many breakthroughs to be made that I think, yeah, but both as a career and as an academic subject to delve into in college, I think there's just so much potential. I think in both in all of your guys' topics, but like, yeah, super, super hot and exciting fields that are kind of exploding right now. How about you, Akshat?
Initially, I was like considering molecular cell biology as like a major in undergrad, but after this project, I'm still on molecular cell biology, but with a focus on genomics. That's definitely like what's changed. Like I definitely like find genomics to be the most interesting part because like it kind of like there's nothing you can't investigate with genomics, so it's really nice.
Totally. And can you tell us what the difference is between genomics and genetics? Because I don't think I'm fully understanding the difference.
Genomics, genetics, I think they go hand in hand. Genomics is more of like, off the top of my head, don't quote me, but like I think it's more of like the whole like genome, like all the genes of an organism. Genetics more specific based, but genomics, genetics, both of those, just something working with DNA.
Got it. Cool. Thank you. Manasvi, any other final thoughts on this question that I threw at you first and then I threw at everyone else?
Not really anything new to add, just that I've been interested in CF for a while. So I think that's the direction I'm just mainly going in for now. And this is just like increased my interest in disease.
Awesome. Super cool. All right. Thank you, everyone. This was really wonderful. I'm glad we had some extra minutes at the end to sort of debrief and reflect on how your Polygens project fits into your larger academic and career thoughts. We can end here a little bit early. Thank you so much. I highly encourage you to continue to hop over to other sessions, learn from your peers. And we're super excited to review all of the presentations and then announce prize winners at the end of next week. So thank you, everyone, and have a great rest of your conference.
I have a question really quick.
Yeah, Krithin.
Are we allowed to like join other sessions?
Oh my God, yes, please. We highly encourage you to join other sessions. There is another round of sessions starting at 11:45. Well, I don't know what time zone you're in, but in 10 minutes. 10 minutes, yeah. So join other sessions. You can just go to the lobby, find the ones that are starting at 11:45 that you're interested in. There's usually split between STEM and humanities and then CS, and then just join them.
Okay, thank you.
All right, see you guys.
Regarding the review of presentations.
Yes.
They'll be going to our presentations and going through them, right? Or
correct.
Okay.
So the judges, if the judge wasn't here listening to the presentation live, then they will review the recordings and then score and then give you feedback afterwards.
Okay, just curious because like I have a lot of skipped slides with like revisions and stuff on my actual document slides. Is the recording all they need, or do they need access to my?
The recording is all they need, but we also have all of your guys' PowerPoints like saved so that they can refer back to it if the slide went really quick or something. So we have all the information we need.
All right, great, thank you.
All right, thanks,
Bye.
team. Have a great
Bye.
conference.